Hi, my project is on deciphering germline mutations from somatic tumor sequencing. Here is my code. Genes are portions of our DNA that control cellular processes. Cancer is fundamentally a disease that is due to alterations in genes called mutations. Some of these alterations are inherited, also known as germline, and the rest, termed somatic, develop during the patient's lifetime. Germline mutations are shared amongst family members and pose increased cancer risk for subsequent generations. Identifying germline alterations helps monitor patients for other cancer risks and alerts other family members to intensify screening for cancers. Recent advances in biotechnology have made sequencing the process of uncovering the exact DNA sequence for genes and cancer tissues obtained from biopsies practical, affordable, and routine. However, inherited mutations are not discoverable by this method that relies on cancer tissues. I sought to develop computing algorithms that would analyze cancer DNA, sequencing for specific patterns, and flag mutations that are likely to be germline. Now, here is my data. As we can see, I have a smaller data set, and I also have a much larger data set. The project came from large dataset tables which contain de-identified retrospective genetic sequencing information from cancer tissues of unique patients. The dataset also contained elements like age, randomly assigned study IDs, and information on the somatic tumor DNA. I partnered with a research collaborator at the University of Michigan, Dr. Anthony Scott, an assistant professor in the Department of Genetics. Working with him, I created this Python-based program called GIA, Genetic Identification Algorithm, to extract genomic information from a database containing de-identified NGS, or data of patients. The information and predicts the type of mutation, that is to say somatic versus germline. It uses for loops, if conditions, and other iteration/linear searches to assign each genomic aberration with a germline or somatic variant label. Using the algorithms, I was able to identify potential germline mutations versus the rest likely to be somatic mutations. I entered all these different types of information into lists and appended them into other data, incorporating the additional subject information such as age and source of the tissue specimen that was sequenced. All the source data sequencing resulted, or in other words, individual cancer DNA sets. Principal among these were the results was the likelihood of a specific mutation becoming germline that I calculated for each alteration. I concluded that the program/code allowed rapid filtration of large datasets and required minimal computing resources. The efficiency of my program was in low runtime and potential high accuracy. In terms of next steps, my next steps would include enlarging the scope and magnitude of this project with larger datasets. It would also be important to confirm GIA in other independent datasets. Further refining the algorithms to increase the probability of germline prediction would also be an important step in this process. Final confirmation of every algorithm generated result is within the scope of my project. I am to continue my project at the University of Michigan, and I look forward to working with them in the future. Thank you.